In this unit, we're going to look at some of the sporozoan organisms. A sporozoan falls in the class Sporozoa. The parasitic protozoa that are sporozoans usually pass through phases of sexual and asexual generation. Typically, they use different hosts, and in one of the hosts, sporogenesis takes place. Organisms in this class include the organisms that cause malaria and toxoplasmosis. Different terminology is currently in use regarding these organisms. We can refer to some of them as apicomplexa. The name of the taxon or grouping apicomplexa derives from two Latin words, apex for top and complexus referring to infolds, and this refers to the set of organelles in the sporozoite. The apicomplexa are a large phylum of parasitic protists. They're unicellular and spore-forming. These organisms comprise the bulk of what used to be called the sporozoa. So as you're studying and in practice, you're going to run into both of these terms. Sometimes the name sporozoa is used as a synonym for the apicomplexa. Occasionally, it's considered a subset. To kind of further confuse things, we have the grouping the Microsporidia, which are a phylum of spore-forming unicellular parasites that were once thought to be included with these protists, but now we think they are fungi. Microsporidia are restricted to animal hosts. All major groups of animals do host various species of Microsporidia. Several species can infect humans. These are most often opportunistic infections. The replication of the Microsporidia takes place within host cells, and the cells become infected by unicellular spores. So let's go back to our original terminology and just think of them as sporozoans. Sporozoans have a life cycle that includes both sexual and asexual phases. Typically, they have two hosts in their life cycle. They produce an infective spore that is ingested or injected, typically by a biting arthropod. One problem we need to be aware of in the lab is that because these are intracellular parasites, when we run them through an automated cell counter, such as a Coulter counter, we'll miss them because they're inside the red cells. And so it's important to know that those differentials are important. The actual looking at red cells and blood smears on a slide so that we can see these organisms within the red cells. The diseases we're going to look at in this unit include malaria, babesiosis, toxoplasmosis, and pneumocystis, or PCP pneumonia. 4 species in the genus Plasmodium cause the disease malaria. And in the Western world, we don't think about malaria very much. Most of us have not personally encountered it. But to give you an idea of how significant this disease is, I went online, did a little bit of recent web research. 2015 was the latest data that I could find readily. In 2015, only a couple of years ago, there were 214 million cases of malaria worldwide, with 438,000 deaths. The disease has a mortality rate of over 60%. In certain parts of the world, especially sub-Saharan Africa, the infant mortality rate due to malaria is exceedingly high. We've worked really hard in recent years to diminish and perhaps eradicate malaria someday, and great strides have been made to lower the numbers drastically. But as you can tell, even as recently as a few years ago, 214 million people contracted it. So we still have a long way to go in eradicating it, if indeed that is ever possible. The life cycle of these organisms is relatively complex, and we'll look at it in a little bit of detail so we can be familiar with the terminology that's in use. This organism is going to have both sexual and asexual phases. The asexual phase occurs in the human being first when the mosquito injects sporozoites into the host. So mosquito bites man. If the mosquito has the organism within it, it shoots these sporozoites, these infective parasitic particles, into the host. Once they're in the host, the sporozoites travel to the liver and invade the hepatic cells. Within those cells, they mature into a form called cryptozoites and continue to reproduce and produce the form called merozoites. So this part of the life cycle of the organism is called the exoerythrocytic cycle because it has taken place outside of the host red cells, and it takes a couple of weeks after the original mosquito bite. So these merozoites that have formed in the liver leave the liver when they're ready, and they enter red cells in the circulation. After they enter the red cells, they become the form trophozoites, and we call those ring forms. And then as the trophs mature, they mature into a form known as schizonts. And in longer-term infections, these schizonts go on to produce more merozoites. And when the merozoites are ready to be released from red cells, they rupture those red cells, and each one is infective and has the ability to invade a brand new red cell. So once these are in the human body, they're reproducing asexually within the red blood cells. As the infection continues, some of the merozoites will become sex cells. The sex cells are referred to as microgametocytes and macrogametocytes. Once the sex cells are formed, then if they are consequently sucked up by a fresh mosquito, the male sex cells and the female sex cells within the mosquito, then sexual reproduction takes place. We're going to use the term paroxysm. And paroxysm is what we observe in the patient periodically who has malaria. The paroxysm occurs when red cells are ruptured. And typically these ruptures happen in mass. And when the red cells rupture, they release not only infective parasitic particles, but they also release toxic products that have been produced in relation to the infection. And so the body basically has an allergic reaction to these toxic materials, and this generates the paroxysm. The paroxysm begins with violent shaking chills, so violent they shake the bed. Probably none of us have had chills like this. And then when the chills subside, the fever follows. And this is a very high fever. Often the person becomes delirious because the fever is so high. So that's the paroxysm, this episode of severe chills and fever. And once the fever subsides, the patient will be relatively asymptomatic until the next paroxysm occurs. Well, it turns out that different species cause a different time lapse between the paroxysms, and this can be used to help in the diagnosis. The true diagnosis is made by the lab identifying the malarial forms in the blood. We want to collect blood between the paroxysms. We don't want to collect it while the cells are lysing, but rather we want to look at the organisms while they're in the red cells. The way that we look at these is to make thick smears and thin smears. If you are an experienced MLT, you know about making thin smears for white cell differentials as a part of the complete blood count. The goal in a thin smear is to spread out those cells so that they're one cell layer thick, so that as you're looking at them and evaluating them, you're looking at one at a time that is separate from its neighbors. A thick smear has the objective of not spreading them out that thin, but rather providing you with lots and lots of red cells to examine in each field of view. They don't need to be separated out and all pretty in terms of red cell morphology. What we're doing is we want to look at lots and lots of red cells to try to determine whether any of them contain the malarial parasite. One thing you'll need to become accustomed to is distinguishing platelets, which are of course very normal in a blood smear, from malarial forms. And so as you look at the picture on the left, there's 3 big black arrows pointing to some beautiful P. falciparum ring forms. And of course there's even more than what the arrows are indicating here. There's several of them. What you'll notice is that there's no halo, there's no clearing around the parasitic infected red cells. If we come over here, this is not Plasmodium, this is not a malaria patient, but what you see here, and here's probably a better one over here, those are just platelets sitting on top of a red cell. And when the platelet is sitting on the red cell, it's sort of pushing down on it just a little bit and squishing it a wee bit. And so it produces that clear area or that halo around it. So you will get lots of practice recognizing what platelets on red cells look like, so that when you get to the point in your career and you happen to be examining a smear for malaria, it'll be very obvious what's a platelet and what is a malarial infected cell. Let's try to work our way through this terminology. And as I go through it, I'm going to give you some of the tricks that I use in my mind to help keep them straight. Some of them might be silly to you, so you may or may not want to use them as you study yourself. So we're going to start up here with the sporozoite. This is the infective form, the spore, if you will. This is what is in the mosquito, such that when the mosquito bites the human, he injects the sporozoites into the new human host. Within the new human host, those sporozoites make a beeline, a straight shot into the liver and become cryptozoites. I remember this one because the medical term crypto refers to hidden. And so the cryptozoites are hidden away, if you will, from the rest of the body. They're hidden or tucked away inside the liver. Now within the liver, those cryptozoites mature to become trophozoites. Now, parasites in general, I associate trophs with being infective. Now, normally my brain is associating trophs and cysts, for example, and the other parasites we've looked at. But the general rule that trophs are infective triggers me to remember that this is what leaves the liver and infects the erythrocytes. And inside the erythrocytes, those trophs often produce ring forms. The merozoites form in the red cells after the trophs have been in there for a while. And merozoites are the developed asexual stage of the sporozoa. Merozoites are released when the red cells rupture. We learned that's during the paroxysm. When they're released, they go on and infect additional red cells and continue on this asexual reproductive stage. Now, the term schizont is basically the developed stage of asexual division. So sometimes the term schizont will be thrown around. If an infection goes on, doesn't get diagnosed and treated, after a period of time, the sexual phase begins, and the sexual phase includes macrogametocytes and microgametocytes. And I keep those straight because macro, of course, means bigger, and the macrogametocytes are the female sexual form, and of course, human eggs are much bigger than their counterpart, the spermatozoa, the male sexual form. Continuing on with our terminology, we're reminded that the paroxysm is the severe chills and fever that occurs in the patient. The paroxysm occurs when the red cells are actively lysing and releasing merozoites and the toxic materials that have been generated. The release of the toxic materials basically causes an immune response that is responsible for the chills and the fever. This paroxysm occurs about every 36 to 72 hours, depending on the species causing the malaria. So as we look at our chart up here, we talk about periodicity, this time between paroxysms. If the paroxysm occurs on day 1, and then again on day 3, and then again on day 5, etc., we refer to that as tertian malaria. And the 3 species that produce tertian malaria are P. vivax, P. ovale, and P. falciparum. On the other hand, the species Plasmodium malariae occurs on days 1, 4, 7, etc. So the doc, of course, can evaluate the timing of these paroxysms and fundamentally have a pretty good idea whether he's dealing with P. malariae or one of the other 3 species, and then we in the lab kind of take over and help differentiate the P. vivax, ovale, and falciparum from one. P. falciparum is the most severe form of malaria. Effects in the red cell that we see is sometimes the red cells are described as being knobby, which may or may not be visible to you as a practicing tech. They also tend to cause the red cells to agglutinate as well as lysing. And as agglutination occurs, of course, these clumps of red cells may plug up small blood vessels and cause issues there with circulation. Because this is so severe and the lysis is more severe with this species, it has a nickname of black water fever. In some cultures, urine is referred to as water, and black water referring to the darkened color of the urine that's produced in these people as a result of this intracellular, or not intracellular, circulating hemolysis that's occurring. P. falciparum invades both young and old mature red cells, and we'll see that the other species are a little different in that respect. And the paroxysms are considered tertian. They occur about every 36 to 48 hours. What we're seeing in this picture is on the left, of course, normal urine. In the middle is a container of urine from a person with Plasmodium falciparum. This is black water urine. Dark, you can see why it's referred to as black water, because there's a lot of hemoglobin in it from all that intravascular lysis that's occurring in the patient. And what they've done in C is just dilute it enough to let you see that it is indeed red hemoglobin when it's not quite as concentrated as what you're seeing in B. Let's look at some of the microscopic forms we may see if we're looking at P. falciparum. Up here, top left, we have some gametocytes, the sexual stage of the organism. You're not going to see these often, but you could. Here we're looking at some very pretty ring forms in the erythrocytes. Over here, we can see, we described that sometimes the red cells look bumpy with this particular organism. Down here on the lower and middle right, we see red cells that are just chocked full of merozoites. These cells are no doubt getting ready to rupture, to release all these merozoites so that each one can go on to infect other red cells. Plasmodium alveoli is the rarest form of malaria. It too has paroxysms approximately every other day, but relapses can come back many years after the initial infection. The trophozoites for Plasmodium alveoli display something called Schüffner's dots in the red cells. They typically invade the reticulocytes, the younger red cells, and when you see an infected red cell with P. ovale, the red cell tends to be a little more oval in shape. Plasmodium ovale invades the reticulocytes. The infected red cells typically are oval. Schüffner's dots are characteristic, and Schüffner's dots were described 100 or so years ago by, I think it was William Schüffner, and that's this kind of stippling effect you see in the red blood cells. And P. ovale is tertian again, meaning paroxysms approximately every other day. Plasmodium malariae has paroxysms about every third day. This organism infects the older red blood cells. When we look at the trophs, the troph may form a characteristic band across the infected red cell. We may see cells with 6 to 12 merozoites present with malarial pigment tucked in there as well. The gametocyte, the sex cell, is ovoid in shape. P. malariae, mature red cells, band-formed trophs, which you can see over here. 6 to 12 merozoites surround dark staining pigment, which you can see down in this photo, and it's described as quartan, meaning every 3 days for paroxysms. Plasmodium vivax is the most widely disseminated form of malaria. People with P. vivax infection can suffer relapses many years after their initial infection. This organism infects the reticulocytes, not the older erythrocytes. An interesting note is that there is a genetic correlation with P. vivax. We know that people who lack the Duffy antigen on their red cells are resistant to P. vivax infection. It appears that there's something with the lack of that antigen prevents the plasmodium from getting into their red cells and causing infection. And so historically, people in parts of the world where malaria was prevalent, there was a high infant and childhood mortality from P. vivax infection, unless they happen to be negative for the Duffy antigen. So people who were negative for Duffy antigen didn't get malaria in their youth, therefore didn't die in youth, and went on to become adults and reproduce. So over the millennia, there's been an evolutionary pressure to have a lot of individuals that lack Duffy antigen in areas of the world where P. vivax is prevalent. So today, about 70% of African Americans are negative for Duffy antigen. Why? Because their ancestors are from part of the world where malaria caused by P. vivax was prevalent. And so there was a pressure on the gene pool that led to the production of many people who lacked that particular antigen. Plasmodium vivax, the most common form of malaria. It only is seen within the reticulocytes, young red cells. Schuffner's dots are visible, 14 to 22 merozoites, and it is tertian. I think it's a very nice thing that in today's world, um, instead of trying to identify ring forms and schizons and merozoites and Schuffner's dots, yada, 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 under the microscope, which is something that some people get very, very good at if they practice in an endemic part of the world. But I think it's kind of nice that today we have PCR to sort these things out and be certain of what we're doing. So just briefly, in lane 1 here on this PCR, this is the band that shows up with P. vivax. Lane 2, the band for P. malariae. Band 3, the band for P. falciparum. And band 4, or lane 4, is the band for P. ovale. So as you can see, given the right lab setup that has the ability to run PCR, suddenly it becomes very easy to speciate an unknown by running the PCR test and comparing with the standards. Babesiosis is an illness that is transmitted by ticks. Ticks are the definitive host for the Babesia species. We are the intermediate host. Most cases of babesiosis, say that 3 times, babesiosis, are actually subclinical, but then, which is a good thing in terms of illness, but it's a bad thing if you're in the blood bank community and you're concerned about the possibility of transmitting this illness from a subclinically infected donor into a recipient and giving them the illness. Symptoms of babesia are similar to malaria, although they're not as synchronized. It's not quite as clockwork-like when they go into the paroxysms, not quite as severe. Symptoms occur, if they're going to occur, a couple, 3 weeks after the tick bite. We have had cases in this country. It states here that the case progression is different in each area, more severe in the northeastern part of the country. Californians usually asymptomatic. Is the organism in the blood, given that he's kind of a third cousin of malaria. Probably that's the first thing that came to mind when you looked at this was, well, it doesn't look a whole lot different than some of the things we were looking at in previous slides. But to review, these are tick-borne parasites. Humans are accidental hosts. We see ring forms only in the red cells. And sometimes they're in pairs and tetrads. So this one looks like a nice tetrad form here. Toxoplasmosis is an illness that many people have heard of, and often they hear toxoplasmosis and they think cats. And indeed, cats are the definitive host for the parasite, although it can infect almost all other animals. I'm going to give you kind of the rest of the story on toxoplasmosis as I understand it, because I think that there's a certain amount of hype that's been given to this organism that maybe it doesn't deserve. You can be the judge. How do cats get the infection? Cats get the infection when they play around with, eat, if you will, mice that are infected. Okay, or from the ingest soil that has been infected. But typically the cat picks up the infectious agent from the mouse. While the cat has the acute infection, which may or may not cause symptomology in the cat, But while they have the acute infection, they'll shed the organism oocysts in their feces, and those oocysts become infective. Most people have, or I should say, 'cause I'm not a cat person, but if you have a cat, and if it's an indoor cat, then it's probably trained to use a litter box, which means that while it has the acute phase of this infection, it's going to be depositing this organism in the litter box. If a human acquires the infection, and they're going to acquire the infection by ingesting the oocysts, most humans are also going to be asymptomatic. So you may have an asymptomatic cat giving this infection to a person who ends up asymptomatic as well. So we never even are aware that it occurs. But the problem is if it's a woman and she happens to be pregnant and she contracts this organism, there can be drastic effects on the child. It may lead to spontaneous abortion, or if the child goes to term, there might be very significant consequences. So here's what has to be aligned in order for this pregnant woman to acquire this infection. Got to have the cat. The cat uses the litter box. The cat acquires the infection during the pregnancy. The woman handles the cat litter. Okay. Maybe personal hygiene was lacking or slipped a little bit, and she, got the organism into her mouth and picked up the infection. That's a bad scenario, but a lot of things have to come together for that. If it's a totally indoor cat, the chances of him getting a mouse that's infected are slim to none, I would speculate. Um, Here's where I'm going with this. I have talked to students over the years, and apparently some docs, some OBGYNs, will flat out tell women, if you're pregnant, you have to get rid of your cat. Um, I think that's a bit of overkill. To my knowledge, the recommendation is that you should probably let hubby or somebody else do the litter box while you're pregnant. Um, and the danger of acquiring this thing is not really that great if you're aware of what causes it and what can prevent it. So that's why I'm throwing that out there, because like I said, I have run into individuals who have basically been forced to get rid of the beloved cat when they became pregnant. So I'm not a doc. I'm not giving you medical advice. I am just trying to kind of fill in some of the behind the rest of the story type information so that you can make your own decision. Now, in today's world, of course, we have a lot of severely immunocompromised individuals. And in the immunocompromised, infection with T. gondii is, of course, a bad scenario. So this slide is showing you the organism in a bronchial alveolar lavage, a lung washing, if you will, that shows the trophozoites of this organism. We will look briefly at a couple of intestinal sporozoans. These are typically associated with causing disease in immunocompromised people, often associated with diarrhea, but they also can be problematic, causing eye infections, keratitis, corneal ulcers in individuals who aren't immunocompromised. So even though the diarrheal aspect we tend to associate with immunocompromised, eye infections due to these organisms are seen occasionally in individuals who have intact immune systems. In the coccidiosporozoans, the infective form is the oocyst, which is passed in the feces of the definitive host. The oocyst contains the sporozoites. After ingestion, the sporozoites are released in the intestine enter the epithelial cells of the newly infected person. Sexual reproduction occurs, resulting in macro and microgametocytes. You're looking at a stained smear here that contains Cryptosporidium parvum. You're looking at the mature oocysts in a fecal specimen. Reminder, this is transmitted by ingestion of those oocysts. In recent years, there have been Cryptosporidium outbreaks associated with public water supplies, unpasteurized apple cider, improperly washed fruits and vegetables, and municipal pools. This organism causes acute diarrhea, which is self-limiting in people with functional immune systems. People who are immunocompromised, it may cause death or it may progress into chronic diarrheal disease. There's been increasing numbers of problems with Cryptosporidium in public water supplies in recent years. If you go to the discussion board, there should be a discussion posted about Cryptosporidium outbreaks. It's for a bonus point on the final exam, so you're not required to do it, but a lot of students find it interesting and choose to do it in order to earn that bonus point and enrich their education a little bit. If you happen to be the first one that goes looking for this and it's not there, just drop me a quick email and I will make it available to you. Isospora belli. There's no known intermediate host. The infected person passes the organism in their stool via oocysts. These oocysts are transparent and immature. They contain a sporoblast, which is a single mass of protoplasm. Within 18 to 36 hours, the sporoblast develops into 2 sporoblasts, and then 4 infective sporozoites are formed in each sporoblast. Full maturation takes 4 to 5 days. Transmission is through contaminated food or water. This parasite is found worldwide. The oocysts stain well with iodine but are difficult to see in PVA or trichrome stained specimens. And here we see 3 images of Isospora belli. Here we're going to introduce ourselves to one species of Microsporidian Sporozoan. The Microsporidia are tissue parasites that are found in immunocompromised patients. They're very small, 1 to about 2.5 micrometers, and they are obligate intracellular parasites. The organism injects the infective contents from the spore into the host cell through a tube. The organism then multiplies by sexual or asexual means, and thick-walled spores are released from the infected cell. Diagnosis is made in histology specimens using acid-fast and PAS stains or alternately electron microscopy. Many Microsporidium species have been identified. The only one we're going to learn is Enterocytozoan Bianusi. It causes a self-limited diarrheal disease that typically resolves in a couple of weeks. We're going to finish up by looking at an organism called Pneumocystis jirovecii. Pneumocystis jirovecii. It previously was known as Pneumocystis carinii. This organism was previously categorized as a parasite. In your textbook, it's still with the parasites, but now the science has moved it into more the yeast realm of life. This organism is the causative agent of atypical interstitial plasma cell pneumonia, abbreviated PCP. This is one of several infections that is almost diagnostic for HIV/AIDS. Essentially, everyone that has progressed into HIV/AIDS will be positive for PCP. And with that, we finish our journey through the parasites. Next week, we'll have the examination on all this material. It's a daunting task. I've been in your shoes. I understand how it feels. Um, do your best. As I said, my favorite tips that work for me in any event is flashcards to study alone with, study with a partner, do some mutual quizzing back and forth, say the words out loud as many times as humanly possible so you become comfortable with them. You make charts. Charts and are often very helpful. You get a lot of charts in the course material, but sometimes making your own charts, organizing things in a chart form that makes sense to you can be very, very helpful as well. So with that, time for me to be quiet so that you can get down to the business of studying.